Okay, so what do you know about microbes already? So I have, I didn't copy it. But let's just go around at your table and just talk to each other for 2 minutes about what does microbe mean to you, and what do you know about a microbe already? What are you scared about? What are you excited about? So just quick discussion about what are microbes in your life. Okay, so I'm hearing I've heard some good stuff there already. I was hearing Aaron talk about bacteria digesting all the stuff. I mean, if it wasn't for bacteria, when we die, nothing would happen we would just stay. So bacteria are the decomposers, along with fungus and other stuff there. So one quick summary from your table, what were you talking about here? So very important in terms of our health, right? And then Aaron's table, I heard him talking about digestion of breakdown of leftover stuff. Okay, how about your table? Your health and that microbes on our skin. Yep. Front table, Catherine, what were you?
It's involved in DNA.
Involved in DNA? Well, you can say the word DNA anytime around me because that's like my favorite thing in the world, DNA. Yeah, so just like we're living and have genes, bacteria and microbes have genes as well. So we'll learn all about the structure of bacteria and their genes and what makes them. And bacteria are actually very simple, but bacteria have been around for 3.5 billion years, which means they've had a lot of time to be perfect. And when you learn about a process called glycolysis, remember glycolysis, cellular respiration? Glycolysis is the first way organisms were able to make energy. So it's a very ancient process. We still do glycolysis in our eukaryotic cells, but so do bacteria. And then we'll spend a lot of time comparing, well, how does a bacteria do cellular respiration, and why does it do it that way? How does bacteria get its food? How do we get our food? We have this amazing mouth and stomach, and we digest it. Well, a bacteria is just a little single-celled organism. It gets its food from where it is, on your piece of chicken, on the floor. It's going to metabolize whatever food source it can, and it's going to grow and grow until it runs out of food. And that's the good news. If bacteria could grow without food, this planet would be just bacteria. So what limits bacteria is growing is really their food source, and that stops them growing. So bacteria have this logarithmic growth when they do grow, and then they very quickly die when they run out of food. But then there's those very clever ones that can form something called endospores and go almost into hibernation and live in a spore form for centuries, millions of years. They actually have pulled out endospores from the pyramids that actually can be viable. So that's the scary part when we start reanimating bacteria that's been in hibernation for a long time. Global warming. Global warming is causing the ice caps to melt, and bacteria and viruses that have been frozen for millions of years are now being put back into the environment. Those are some scary things. It's very interesting when you start looking at our little friends. So just in terms of microbes in our life, a few are pathogenic. Not many bacteria are pathogenic. Pathogenic means, the word pathogen means to cause disease. So pathogen causes disease. Microbes decompose all organic waste. What do you think happens in garbage heaps when we send stuff to the garbage heap? All of our sewerage goes to a place where microbes are involved in decomposing it back into its basic building blocks. The producers in the ecosystem by photosynthesis. Everybody always thinks, oh, we get all this wonderful oxygen from the trees. But bacteria was around a long time before the trees. The first organisms to do photosynthesis were bacteria. And plants came much later in evolution. So the first photosynthetic organisms that figured out to use the energy from light were bacteria. And in fact, they didn't use red and blue light. They actually used green light. So photosynthetic bacteria actually use green light, whereas photosynthetic plants, we use the blue light and the red light, the other end of the spectrum, because that was the light that wasn't being used up by the photosynthetic bacteria. So some bacteria use that spectrum, and that would be the line that eventually became the chloroplasts that landed up in the cells. A lot of our industrial chemicals, such as ethanol and acetone, are actually produced by bacteria. Anytime you're eating fermented food, vinegar, cheese, bread, you're using a microorganism. What microorganism do we use to make bread? Yeast. So yeast is a microorganism. Yeast can be harmful when it outgrows and you get yeast infections. You might have heard of that. And then many products in manufacturing, such as cellulase to help break down cellulose. There are some products like in jeans, they use bacteria to make the fiber of the material softer. Stonewashed jeans are due to bacteria. And then, of course, disease treatment, things like insulin. Insulin is due to recombinant DNA, and they've actually made human insulin in large amounts in bacteria. They've taken that gene, put it into bacteria, so we'll learn about that, how you can make human insulin, and they produce vast quantities. 50 years ago, people who were diabetic used pig insulin or bovine insulin, and it wasn't a perfect match, but it was better than nothing. Today, people who are diabetic get human insulin, but it's human insulin not extracted from other humans, but where the gene is actually used to produce the insulin in a bacteria. So here's some of those designer genes made by microbes. These are some names of bacteria. Stonewashed genes. They used to the reason they were called stonewashed is because they used to take the fabric and wash it with stones to make the fabric softer. And then they figured out that this bacteria produced an enzyme that softened up the genes. And then they didn't have to spend all that time rinsing out the grit from the fabric before they could make jeans, because no one would want to walk around in crunchy jeans, right, with bits of stone in them. So you've just looked down the list. Bleaching using peroxidase from a mushroom. Indigo genes, the color indigo, it's one of the metabolic process of E. coli that can actually produce that indigo color. And so that is actually a color that comes from bacteria. So they really are our friends. And then, of course, bacteria can also metabolize plastic, which is pretty cool for breaking down some waste, and that's probably going to become more and more important as we fill up our planet with a lot of junk. Okay, just some quick review, naming and classifying microorganisms. We will spend chapter 10 talking about that, but it's all based on Linnaeus' system of scientific nomenclature, where when you get down to the end of that whole diagnosis, you have a genus and a species. So all bacteria you're going to learn are pretty much going to have a genus and a species. And when you write these names, they are usually italicized or underlined. So if you write a bacteria name, you should actually underline it. Not everybody does that, and I don't enforce it, but we really should be doing it. And the other thing is the genus is always capitalized, and the species is lowercase. And of course, the names of bacteria will drive you nuts because of how they decided to name them. They're Latin names, many of them, and they basically Latinize of a name that they decide to give the bacteria. And they're used worldwide. And many of these names honor the scientists who found them, and that's why they have such really funky names. And sometimes the names are a bit simpler because of the place they occur on the body. So, microbes in our life. We will learn all about microbes and how to prevent their growth. So, knowledge of microbes allows us to prevent food spoilage. Anyone know about how much food actually gets wasted because of bacteria spoilage? Probably around 25% of food actually gets ruined. Think of your own refrigerator. How often are you throwing some stuff out that you made, that you bought, and you didn't consume, and now it's spoiled? And that's because of bacteria growth on it. Knowledge also has helped us to prevent diseases. And we'll talk a little bit about history at the end of today of how that whole evolution happened when they understood microbes cause disease and how we lead to modern medicine today. Aseptic techniques. Most of you do some aseptic techniques all the time. You wash your hands, hopefully before you eat, and hopefully every time you've been to the bathroom. That's just normal, good hygiene. Anyone know what the #1 advance is in the world in the last 150 years? What has been the biggest advance to actually save human lives? If you would have to guess. Penicillin? So antibiotics, you would think that would it? That's a good guess. It's not correct. Anybody else?
Soap.
Sorry?
Soap in general.
Not even soap. Clean water. Clean water and sanitation is the biggest change in evolution of human in their development for healthier people. So when you think of all of the developing world, if we can just, instead of like sending all the vaccines and the antibiotics, which they need, but if we start off by first giving them a better infrastructure and having those people have access to clean water and sanitation, that they don't go to the bathroom where they get the food that they eat and wash their clothes from. That is the huge thing, because that is the source of infection and contamination. So when you look back in 300 years ago, how people lived to how we live today, I mean, just having a flushing toilet in your house is a fantastic thing. You go to places in the world where they don't have that, and you realize what a luxury it is. And we just, in this country, we take it for granted. It's a wonderful thing. So we'll talk about aseptic techniques that are used all the time, and you'll learn how to be aseptic in this lab. So here's just showing some bacteria, some common names. Everybody knows the name E. coli, right? But E. coli actually honors the discoverer, Theodore Escherichia, and it's a kind of a name that I always get tongue-tied on. But Escherichia coli is the most common bacteria that lives in our colon, and it keeps us healthy. And when you will learn, this is one of the bacteria that we use in the lab all the time. When you look at it under a microscope, you'll discover it has this very specific rod shape. It's a small rod-shaped organism. If you had a way to stain it, you'll discover that it has these lots and lots of flagella. So it's got flagella all over it. So E. coli can swim and run and tumble and get to places it shouldn't get to. So the most common bacteria in UTI infections, urinary tract infections, is actually E. coli, because it's the exit point from our body in our stool is very close to our urinary tract, and then often that just cross-contamination from unhealthy habits is getting E. coli in the wrong place. So when you write and coli, it comes from the species name coli comes because it's most common in the colon. So a lot of bacteria, there's a little bit about where they come from, but again, a lot of the names just do not make sense. Here's some pictures of Staph aureus. Staph aureus was named for its golden color. So aureus comes from meaning gold, and staph comes from the way it actually exists. So staphylo is when you have clusters or clumps. So they look like bunches of grapes. This is us scanning the electron microscope where you've seen that three-dimensional structure. This is what we will, if we were looking at staph under a microscope. We do not use Staphylococcus aureus in our labs because it's an opportunistic pathogen. It can become a pathogen. So we will use Staphylococcus epidermidis, which is another of the staph family. So you will see it look exactly like that, but it's not aureus. And that's one thing that you'll need to do in your lab book, is always change the name. And in my final practical, one of the questions was for a bonus. It said, name 3 bacteria that we used in this class. And more than 50% of students wrote Staphylococcus aureus. When I told them from the first day, it's not a bacteria we'll use in this class because it's an opportunistic pathogen. So don't get that wrong. Staph aureus is the one that's causing all the MRSA problems. Any of you work in hospitals? Heard of MRSA? So MRSA is methicillin-resistant Staphylococcus aureus. It's a strain of Staph aureus that is resistant to almost every single antibiotic that's out there. And that's the scary thing for a patient when you get an infection with it. I think there's down to 1 or 2 antibiotics. If you watch the news, you'll realize how this is becoming a problem. We're running out of antibiotics. Antibiotics were the miracle drug of the 20th century. And it's not going to be the same way in the next 50 years. So just naming bacteria, make sure when you name them, you always use the scientific names And the first time you write a bacteria name out, you'll see whenever you're reading literature, the first time they have to spell it all the way out. Escherichia coli, Staphylococcus aureus, and then after that, they can then abbreviate it with just E. coli or S. aureus. But the first time that bacteria is mentioned, they have to have the full name out. And that's because there are other bacteria that can be abbreviated E. coli, but the genus is not Escherichia. It's another version. And here's my take on bacteria names. I don't expect you to learn all of the bacteria names you're going to come across, but I do expect you to learn the names of the common ones that we talk about a lot, like especially our lab friends, the bacteria we're going to use in this lab. Staph epidermidis, E. coli, Bacillus megaterium, Serratia marcescens, all of those. And I can say these words easily now, but I remember the first time I was teaching these bacteria names, and it's hard. But get familiar with them. Make a plan to say a bacteria name to your lab buddies every single day so you get to know them and get named, especially when we do the lab portion. And a lot of the bacteria that we're going to actually grow up today in our plates that we find around campus are probably going to be of the Staph family. And you're going to see some other stuff. We should not grow up E. coli. And there are special ways to grow up E. coli. E. coli, because it's the most common bacteria found in our colon, it's basically an indicator bacteria. So when they are looking for like broken water mains, the people who do water testing, if you work for the county health, they go test water around the county all the time. They're looking for E. coli because E. coli is an indicator that it's been contaminated with human sewage or contaminated with fecal matter, even from animals. So it's an indicator. And then you get the boil water alerts. Because if there's E. coli in the water, then there could be other harmful bacteria that could be contaminated that in someone who has infection, say like cholera or something like that. So that's all those anytime we have broken water mains, boil water alerts, just because you could get contamination. In your textbook, there's a nice table with scientific names and pronunciations, so have some fun trying to figure out those, depending where they come from. So you mentioned penicillin. Penicillin actually is extracted from a fungus, and the fungus name was Penicillium chrysogenum, and penicillin is actually a product of that fungus. It makes an antibiotic to kill bacteria so the bacteria doesn't inhibit its growth. That's what antibiotics are. They're naturally made products by bacteria to inhibit the growth of other organisms so that they can survive. It's kind of like a defense mechanism. And we have figured that out, and then we just extract that antibiotic from that bacteria. So when we do a section on antibiotics, you'll actually see the names of all the bacteria that we extract antibiotics from. When you go to the doctor and you get antibiotics, most of the time they're not really antibiotics, they're antimicrobials. They're synthetic versions of what a bacteria would make. And so we loosely call them antibiotics, but it's really antimicrobial, against microbe. And true antibiotics are made by a microbe against another microbe.